It's 10 o'clock, so we'll get started. Good morning, everyone. I would like to welcome you all, faculty, and family and friends, for attending Mary's seminar today. I'm very happy to introduce Mary Steele, my graduate student, Dr. Hans's graduate student, in the crop science specialization in the plant sciences department, who conducted research at the Cal Poly Strawberry Center. Mary studied at University of New Haven, Connecticut, and then transferred to Cal Poly to conduct her bachelor's degree in biological sciences with a minor in Spanish. And she started her master's thesis in the fall of 2021. And she was co-advised by me and Dr. Gerald Holmes. Very interesting fact is that she conducted 2 projects that we didn't even have a graduate student on at the beginning. So you can be the judge how well we could have handled these projects without her. She was very persistent, so we recruited her. I think it was one of the wisest decisions we took. When it comes to accomplishing goals, Mary goes above and beyond. For example, she published her first paper in Plant Health Progress early this year, and she's ready to submit the next one. She was admitted to the Plant Path program at Washington State University and will be heading to start her PhD this fall. She's even leaving long-lasting memory for us. He has prepared his handbook for graduate students that's going to help us as well as our future graduate students. Without further delay, I would like to invite Mary Steele to share her findings.
Good morning, everyone. Thank you for the warm introduction. So my name is Mary Steele, and I am pursuing a master's in agriculture with a specialization in crop science. And both of my thesis projects center around soil-borne pathogens of strawberry in the Central Coast region of California, more specifically surveying them and cover cropping with wheat for management of Macrophyllina phaseolina in particular. On my graduate committee is my advisor, Dr. Shashika Hewavitharana, and also on my committee are Dr. Gerald Holmes and Dr. Dan Lagarde. And I'd first like to acknowledge and thank all 3 of them for their guidance and support, both relating to the project and otherwise. And I can confidently say that I wouldn't have the future that I have were it not for all 3 of them. I'd also like to thank all the help I've had that has made these projects logistically possible. So my fellow graduate students at the Strawberry Center have provided a lot of companionship to me, as well as members from the Strawberry Commission and undergraduate students. I'd also like to thank our lab manager, Vivian Longacre, and our farm manager, Drew Summerfield, and finally our field research manager, Kyle Blauer, for their help. Funding for these projects came from the California Strawberry Commission, and so I'd like to thank them for their funding support and their help along the way. Dr. Peter Henry and Polly Goldman were collaborators with the Watsonville Salinas survey, as well as members from the industry. Dr. Eric Brennan, Dr. Charlotte DeCock, and Dr. Mark Mazzola all assisted with their expertise on the wheat cover cropping project, and so I'd like to thank them as well. So before we get started, I'd like to ask you all a question, and I think it'll be a pretty easy one. Which field would you prefer if you were a strawberry grower? And particularly if, as is the case for most growers, if your livelihood depended on it. So for me as a pathologist, I have to say that the field on the right is more interesting, but I think the difference that we see here between a field that is impacted by soil-borne pathogens and one that's not, probably, is really big, and that is the reason for my research the past 2 years. Today, I'm going to first give an introduction. I will then go into discussing the soil-borne pathogen surveys I conducted in Watsonville, Salinas, and Santa Maria, and I will then go into my second project, which is looking at the effect of wheat cover crop on the soil microbiome and Macrophyna root rot of strawberry. So first, some introduction. 20% of the world's strawberry production happens in the United States, and within that, almost 90% happens in the state of California. There's a pretty narrow range where strawberries are produced within the state. As you can see, all strawberry production falls in the Central Coast region. We have 3 districts of strawberry production in the state: Watsonville, Salinas, Santa Maria, and Ventura, Oxnard. If we think about this relatively small area, we can compare it to a crop like almonds, which cover much more area and bring in about $11 billion every year. And we think about this crop covering 400,000 hectares. Compared to strawberries, this crop brings in 3 billion every year, but only covers about 12,000 hectares. So if you think about the value of this crop, each strawberry plant counts and is very valuable to strawberry growers. Typically, in the state of California, growers follow this growing timeline for the season. They plant bare root strawberry transplants, which come from nurseries, in the fall. These transplants then have time to experience chill hours over the winter months. That way, in the spring, when warm weather comes, they are ready to start bearing fruit. And this fruit is harvested throughout the rest of the summer. Some strawberry growers in California take advantage of warmer weather, particularly in Santa Maria and Ventura, Oxnard, where the climate allows for it. And they have an alternative strategy, which is planting in the summer and then harvesting fruit even later into the summer, fall, and winter. And as you could imagine, the winter is a really opportune market, as there isn't as much production from other California districts. So I'm just pointing out that these are 2 strategies that growers use, primarily this fall planting strategy, but we also see some summer plantings. Soilborne pathogens are the reason I'm here today, and 2 of them have been around in the strawberry industry in California since as early as the 1960s. These are Verticillium dahliae, which is a fungal pathogen that causes verticillium wilt, and Phytophthora species, which is a genus of oomycete pathogens, which are close relatives to fungi, which cause Phytophthora crown and root rot. And so quickly, just to clarify, when I say soilborne, I mean that the pathogen lives, disperses, and survives and reproduces in the soil rather than above the ground, like with some other pathogens. Methyl bromide was a very common pre-plant soil fumigant that was used widely and was very effective against soilborne pathogens. However, due to human and environmental impacts, it was outlawed through the Montreal Protocol. And for California strawberries in particular, it was banned between 2005 and 2016. So after 2016, this fumigant could no longer be used in California strawberry, for the most part. And after that, 2 additional pathogens emerged in California strawberry. These are Macrophyna phaseolina, which causes Macrophyna root rot, and Fusarium oxysporum formispecialis frugariae, which causes Fusarium wilt. And I've shown some of the survival and reproductive structures of these pathogens here. All 4 of these pathogens cause symptoms in strawberry plants, such as wilting and eventually complete plant collapse. So you can't tell apart the symptoms of one of these pathogens from the other. It's very difficult, but they all cause severe loss, and you can imagine how this might affect yield. I'm going to go into Macrophyna in particular for just a moment, as it is the focus of my second project. So it's one of these 4 major pathogens. As I mentioned, it's a fungus. More specifically, it's in the phylum of Ascomycota and the family of Botryospheriaceae. It is known to have 71 hosts, so it is known to have at least 71 plant species that it can successfully infect and cause disease in. Its optimal temperature is a bit warmer than room temperature, so it prefers 30 to 35 degrees Celsius. It also prefers dry soil conditions, so below 60% soil moisture is optimum for this pathogen. What's so tricky about this pathogen to manage is it produces microsclerotia, which are dense masses of mycelia that can survive for up to 15 years in the soil without a host. So even if you didn't plant a susceptible host for a long amount of time, these structures are still able to persist in the soil for later infection. Here a picture of microsclerotia shown under the compound microscope. And here's just an example of the die-out that this pathogen can cause. What options do growers have currently for management? The first line of defense is cultivar resistance. So if you have a strawberry cultivar that's resistant to a pathogen, that's one option that growers have. This is most common for Fusarium, Oxysporum, Formospecialis frugariae, as there is a gene that has been identified and is now being bred at UC Davis into strawberry cultivars and is somewhat widely used. For the other three pathogens, there hasn't been as much success with developing resistant cultivars. There are some cultivars that are at least partially resistant, but these cultivars are not really widely grown, as there are lots of other factors for growers to consider when choosing a cultivar like yield. Pre-plant soil fumigation is another option, and this is used very widely by conventional strawberry growers. So this is not an option for organic growers, but this involves injecting a gas such as chloropicrin into the soil under an impermeable tarp before planting strawberries. So that's an option that is very widely used for conventional growers. Additionally, crop rotation and cover crops are employed. So crop rotation is when a crop other than strawberries is employed part of the year. And these crops are grown for cash. So you can imagine something like broccoli that they grow, and then also that brings in an additional profit. Cover crops, on the other hand, are not grown for profit. They're just grown for some other benefit, like soil health. Steam is also being investigated as an alternative for organic growers to do pre-planting to kill microorganisms in the soil. Anaerobic soil disinfestation is also being investigated for organic growers. This involves creating complete anaerobic conditions in the soil, which can then kill microorganisms. And crop termination is also being investigated, which is injecting what would usually be used as a pre-plant fumigant, but injecting it into the drip system at the end of the strawberry growing season. When you're done harvesting, it will kill the strawberry plants, but it will also kill any pathogen that has infected and gone inside the plant. And so this is another option that's being investigated. So if any of these were completely effective, I wouldn't be here. So I'm here to investigate an alternative in particular to one of these, which is looking more closely into cover cropping. But these are the strategies that growers have as of now. That brings me to my first project, which is soilborne pathogen surveys in California growing districts, so Watsonville, Salinas, and Santa Maria. And our question was, we know that these 4 pathogens are now the most common and prevalent pathogens in California strawberry. However, which one of these is more common than the other? How is their relative prevalence in the different growing districts? And there's a lot of speculation about this. And that speculation, when talking to growers and hearing from people who are out in the fields, led us to think that maybe since the phase out of methyl bromide, Fusarium has become the most common soilborne pathogen in Watsonville, Salinas. That was something that some growers were thinking about up there. Additionally, we have a diagnostic service here at Cal Poly where growers can send in plant samples and we do diagnostics. And we had been getting a lot of samples from Santa Maria that were positive for Macrophamina faecelina. So we thought maybe this one is most common in Santa Maria, but this had never been actually studied. So we wanted to actually go out and quantify which one is most prevalent. The first survey was conducted in Watsonville, Salinas between August and November of 2021. I collected 74 samples from 69 fields, and this difference is because some fields had some element like more than one cultivar or some growing practice that we wanted to sample as 2 different samples. The samples that we collected comprise about 33% of the district hectareage of strawberries that were planted that year. The second survey occurred in Santa Maria between June and October of 2022. We collected 100 samples from 68 fields. And as I mentioned, in Santa Maria, they employ both fall and some summer plantings. So we sampled both. So we sampled any field that had plant die out. And so the fields that we sampled represented about 30% of the fall planted hectareage and 14% of summer planted hectareage of strawberries that year. Finally, the third district, there's a survey being conducted by another graduate student at the Strawberry Center, Samantha Simard, and that is ongoing in Ventura Oxnard. For each field we visited, the first thing we did was take field observations. So we noted various field variables about the field. We then collected our plant sample, and each plant sample consisted of 8 symptomatic strawberry plants. And we went to the area of the field that showed the most die out, so the area of the field that looked the worst. But then within that area, we tried to collect plants from different beds and from different areas of those beds. We then processed those plant samples, and we got those down to the crown tissue, which is that woody center part of the strawberry plant. We extracted DNA from that crown tissue, and on those DNA extractions, we performed various pathogen diagnostics, which I'll go into in just a minute. Once we had our results, we also made a grower report to send back to the grower who allowed us to sample their field so that they could know what we had found. We had 2 diagnostic methods we used. The first was molecular. So the first thing we did was extract DNA from crown tissue, and then we performed recombinase polymerase amplification for each of the 4 pathogens separately. And then we looked at what result we got. So this is an example of what we would regard as a positive result, because we see a successful extraction, as we see the green line, which represents plant DNA, which we would expect from a strawberry plant sample. And we also see the blue line, which is representing the detection of whatever pathogen this test was for. And so we would call this a successful and positive result. If RPA, or recombinase polymerase amplification, was negative for all 4 pathogens, we went a second route. And this was to plate various plant material, roots, crowns, and petioles onto various semi-selective media, and then saw if anything grew. So we were verifying the absence of those big 4 pathogens and also seeing if there was additional pathogenic fungi. We also wanted to compare various field variables to the pathogen outcomes. So we collected information when we were in each field, such as location, number of drip irrigation lines that a grower used per bed, whether a field was conventional or organic, what the previous crop was there on that same piece of land. So was it strawberries or was it something else the year before, and what soil type. We performed nominal logistic regression on these field variables, comparing them to whether or not the sample was positive for each of those 4 pathogens. And we use standard least squares method to compare the field variables to disease incidence in the field, which is a measure of the percentage of plants in the sampling location that were at least 50% necrotic. So basically how bad was the outbreak there, how many plants were dying. And this was all done in JUMP statistical software. This is a map of the fields that we sampled in Watsonville-Salinas in 2021. So dark gray plots are strawberry fields that we sampled. The light gray is additional strawberry fields that year that we did not sample. And as I mentioned, the sample fields represents about 1/3 of the acreage that year. In Watsonville-Salinas, we found that there was at least 1 pathogen of the big 4 in about 75% of samples. So this left 25% that weren't positive for any of the 4. And we only visited fields that had evidence of plant die out. So this was an interesting finding for us, that not all of them were positive for one of the big 4. Within the 4 pathogens, though, there was relatively equal prevalence. So all 4 major pathogens were found in between 22 and 31% of plant samples. So we can conclude here that all 4 pathogens were similar in prevalence in Watsonville-Salinas in 2021. We also saw that disease incidence was higher on average in organic fields than conventional fields, although there was a similar proportion of conventional and organic fields that were positive for at least 1 pathogen. So in both conventional and organic fields, 75% of fields approximately were positive for at least 1 pathogen. So conventional and organic fields are both experiencing losses from soil-borne pathogens. In Santa Maria, as I mentioned, we sampled both fall planted and summer planted fields. So I've depicted the fall plantings that we sampled in red, the summer plantings we sampled in yellow, and all other strawberry ranches that year in 2022 in gray. And I'm going to show you just a closer look at one of those areas. So you can see the red fall plantings that we sampled, yellow, summer, and then the gray is additional strawberry ranches. In Santa Maria, again, we saw about 80% of samples were positive for at least one pathogen. However, the pie chart looks a little different here. So we saw 52% of samples were positive for Macrophyllina faecelina, whereas in Watsonville, there was relatively equal prevalence. So Macrophyllina faecelina was the most common soil-borne pathogen in Santa Maria in 2022. So why is that? Why would we see much more Macrophyllina in Santa Maria than Watsonville Salinas? We saw that within the district, when we compared the field variables to the disease, to the pathogen outcome, we saw that this pathogen in particular was more likely as longitude decreased. So that means more eastern. And as you can imagine, that would be further away from the ocean where there might be less temperature regulation and highs might be higher. We also saw this pathogen four times more likely in samples that had a back-to-back strawberry planting. So we asked the grower, did you plant strawberries here a year before? Or was it something else? Was it like vegetables? And we saw this pathogen was four times more likely where they had planted back-to-back strawberry. And this makes sense to us as pathologists because the level of pathogen that can be found inside a strawberry plant can be up to 1,000 times higher than that's just in the soil. And so if you were to incorporate a strawberry crop from the year before back into the soil, that would amplify the amount of pathogen for the next year. Another thing to look at with the difference in prevalence in this pathogen between Santa Maria and Watsonville is the temperature. And so we saw that in Watsonville Salinas, it's cooler on average. We also saw that samples in Watsonville are closer on average to the ocean, which we know could contribute to temperature regulation. And another thing that was interesting for us to look at is how irrigation practices might have influenced the prevalence of this pathogen. So in Watsonville, the grower standard is to use 2 rows of strawberry plants per bed and 2 rows of drip lines. However, in Santa Maria, growers usually employ 4 or sometimes 3 rows of plants and then 3 or sometimes 2 rows of drip line. So if you think of the ratio of rows of plants to drip lines, in Watsonville Salinas, it's always 2 to 2. But in Santa Maria, that varies. And so the ratio could be higher on average, meaning that each plant may be getting a different water distribution or maybe even a lower water distribution than it would be in Watsonville. Interesting thing that we saw in Santa Maria that's worth mentioning, it was a friend that we did not expect to see, and that is Meloidogyne species, which is a genus of root knot nematodes. And I've shown here this bottom left picture is the nematode itself under the compound microscope, but they're microscopic, so you won't be able to see them with the naked eye. But what they cause is root galling on the, swollen roots on the plants. And although they don't cause as much significant symptoms as the other soil-borne pathogens I've mentioned, they can cause stunting and yellowing of leaves, which is enough to cause a yield loss. And so we detected this in 6 samples in Santa Maria from 4 fields. And in each case, there was also another of those soil-borne pathogens present in the same sample. These samples that were positive for Meloidogyne had a patchy distribution throughout the district, so they weren't just clustered in one area in particular. We sequenced 4 of these 6. We sent them out for sequencing at the CDFA, and that came back as Meloidogyne hapla. And although on its own, this pathogen doesn't cause really significant loss usually, when it occurs with another pathogen, it can, especially with FOF, which I've abbreviated, and that's the Fusarium oxysporum forma specialis frugariae. And when it's found with that pathogen, it can inhibit the resistance mechanism in a strawberry plant. So if a grower had a strawberry cultivar that's normally resistant to Fusarium, if there's Meloidogyne in the field, there's something between that interaction that can cancel out the resistance in the strawberry plant. So now you not only have root knot nematode, but you have the symptoms from Fusarium potentially as well. So I'm going to take a little pause here, and then we'll move on to looking at the effect of wheat cover crop on soil microbiome and Macrophyllina root rot of strawberry. Okay. So why did we choose wheat as a cover crop to look at in terms of management for Macrophyllina? Wheat has been shown to be associated with the genus of bacteria, the Pseudomonas genus, which these bacteria are not pathogenic to strawberry or to wheat, but have been connected to certain cultivars of wheat, as there may be something in the root system that allows the amount of this genus to be amplified in the soil. This genus of bacteria has also been shown to potentially antagonize various soil-borne fungal and oomycete pathogens. And so our hypothesis was maybe this genus of bacteria could mitigate Macrophyllina phaseolina, a soil-borne fungal pathogen, in the soil, and thus potentially reduce the disease we saw caused by the pathogen in strawberry. So what's different here compared to previous research that has been done with wheat is we were conducting this in a field setting, and we were also using wheat cultivars that are commonly grown in California, whereas a lot of previous research had been done with Washington state cultivars. The objectives of the project were to ask the question, can wheat as a cover crop aid in management of Macrophyllina phaseolina in strawberry? We also wanted to look at how the soil microbiome changes with wheat. So how does the composition of both fungi and bacteria as a whole change over time? Whether there is something in particular that we should be focusing on there. To accomplish these objectives, we conducted a replicated greenhouse trial from fall of 2022 to spring of 2023, and a replicated field trial between fall of 2021 and summer of 2023. So the second field trial is still ongoing. The experimental design for the greenhouse trials consisted of two different factors. So not only did we want to look at various cover crop cultivars, we also wanted to look at any additional effects from pasteurization. And pasteurization we used as a way to kill microorganisms in the soil, as we couldn't feasibly use fumigation in a greenhouse trial. This served as an alternative way to accomplish the same thing. We used 4 cover crop cultivars. We used 3 wheat cultivars, and we used a rye cultivar as well, just because rye is something that's already used in strawberry production or in sort of crop rotation systems in California. So if the same effects were seen in rye, that would be logistically possibly easier for growers to use. We also had a no cover crop treatment. And you see that some of these little pots here are going to be pasteurized and some are not. So we combined all of those, either one of the cultivars and then either pasteurized or not, into giving us our treatment. So we had 10 different treatments, and each trial had 5 blocks. First, we had to inoculate the soil in all the pots so that it had our pathogen that we wanted to target. And so we made cornmeal sand inoculum of Macrophyllina phaseolina, and we then inoculated that into the soil so that the soil was at a level of 2 colony-forming units, or CFUs, which is sort of the amount of pathogen in the soil, 2 CFUs per gram. And we know this level is enough to cause symptom expression in strawberry plants. We let the inoculum colonize the soil for 2 weeks, and then we either seeded the pots with one of those 4 cultivars of cover crop, or we did not. So we also had a no cover crop treatment. This wheat rye grew for 90 days, at which point we chose to not incorporate the wheat or rye shoots. So we cut those off, but then incorporated the stubble and root material into the pots. And we then either pasteurized or did not each pot. And those pots that were designated for pasteurization the soil from those pots went into the oven twice, each time for 8 hours at 80 degrees Celsius. We took soil samples at this time as it could be considered post-treatment. And then we planted 1 strawberry transplant into each pot. And we used the cultivar Albion, as we know it's very susceptible to Macrophromina faecelina. These strawberry plants grew in every pot for 5.5 months. And during that time, we monitored the plants for mortality so we can calculate disease incidence and area under the disease progress curve, which is just a measure of how many plants are dying over time. With the soil samples, we wanted to see if there was any impact from any of the treatments on the level of pathogen in the soil. So to do this, for each soil sample, we blended the soil with bleach or 1% sodium hypochlorite. We then put it through a very fine sieve, and we poured that fine soil directly into media before we poured it, so that once the plates hardened, the soil that was in the media would possibly elicit the pathogen. And so we counted how many colony-forming units or CFUs we saw on each plate. And this red circle is showing you what we would designate as 1 colony-forming unit, as it originated from 1 single microsclerotium of Macrophromina. And from these numbers, we could calculate an overall CFU per gram for each sample. So results from the greenhouse trial. This is showing you AUDPC, or area under the disease progress curve, which is just a measure of disease incidence over time. So we did not see a significant reduction in disease between the non-cover cropped pots or any of the 4 cover crop cultivars. However, we saw lower AUDPC or disease in the pasteurized pots compared to the non-pasteurized pots. And this is for trial 1. We saw very similar results in the second trial as well. So from these, we can conclude that pasteurization reduced disease in the greenhouse trial setting, but wheat and rye did not. How about the pathogen itself in the soil? Again, we did not see significant difference in pathogen levels in terms of CFU per gram between any of the cover crop treatments and the no treatment control. However, we did see a significant reduction in CFUs when we pasteurized the soil versus when we did not, which is what we expected. Very similar results from trial 2. So we can conclude that pasteurization reduced pathogen inoculum in the soil, but wheat and rye did not in this setting. Moving on to the field trial. So this happened, these are happening in Cal Poly Field 35, which is, you can see from your seat, it's right out there. And the first field trial occurred between October 2021 and August of 2022. And we had 4 treatments. So we had a preliminary greenhouse trial that allowed us to narrow down to just 2 wheat cultivars we wanted to select for the field trial. And these were selected based on the cultivars that had elicited the most Pseudomonas bacteria. And so these 2 cultivars were WB 9229 and Summit 515, which are both hard red spring wheat cultivars. We also had a fumigated control, and these plots were fumigated sort of to mimic like a standard grower practice that I mentioned earlier. These are fumigated through the pre-existing drip irrigation lines with maximum rate chloropicrin or trichlor EC. We also had a no treatment control. The first year, we had 4 blocks following a randomized complete block design. And we know that the Cal Poly Field 35 has a high pre-established Macrophromina faecelina population, because in previous years, we've conducted trials there that have inoculated the soil artificially. So we didn't further inoculate for this trial. The second year, same treatments. We just had 6 blocks in a slightly different layout. And this is again in Cal Poly Field 35, where we have an established pathogen population. For each field trial, first thing we did is we had to get creative because we wanted to follow a randomized complete block design, where you could imagine a grower might grow wheat just flat in the field. We couldn't do that. We had to plant wheat directly into the already raised strawberry beds. So we cut open the plastic mulch where wheat was going to be planted, and we then seeded wheat and grew it from about November to January. And before we had planted wheat, we collected a soil sample, and this was our pre-treatment soil sample. And we then, after the wheat grew so that it was in flower, we decided to cut it down, and we did not incorporate the shoots into the soil, but we did incorporate wheat stubble and root material with the rototiller, as we thought that's where the microbiome activity was happening. And at this time, we also collected a post-treatment soil sample. The fumigation during both years occurred in October in the designated plots, and the no treatment control plots had just been sitting covered with plastic during this time. At this point, we were ready to plant Albion strawberry transplants into all plots. So we planted 3 rows of Albion strawberry plants, and we had 3 drip irrigation lines per bed. We know, as I mentioned earlier, that Albion is susceptible to macrophamina, and these strawberry plants grew until August. And throughout the season, we monitored them for plant mortality so that we could calculate disease incidence and area under the disease progress curve. Finally, we collected an end-of-season soil sample. This time, we sampled from the rhizosphere. So that's the soil that's right up next to the strawberry roots. And so I'll mention this as I discuss results, as it makes the results just slightly different from collecting bulk soil as we had previously during the pre-treatment and post-treatment samples. With every soil sample we collected, both pre-treatment, post-treatment, and end of season, we quantified macrophamina, same way as we did with the greenhouse trial. We also, with the field trial samples, we extracted DNA from all samples so that we could send these extractions to a third-party lab for Illumina sequencing of 16S and ITS genes. So the 16S gene is a target for bacteria, and ITS is a target for fungi. And there's a lot of applications you can do with sequence analysis. And so I'll go into just a few of the analyses that we conducted for this trial. The data analysis for the sequence results were conducted in explicit for alpha, beta diversity, and 2-part analysis. We also used PAST for non-metric MDS or multidimensional scaling, as well as unweighted pair group method with arithmetic mean or UPGMA. In year 1, we saw that fumigation significantly reduced disease in strawberries from Macrophamina faecelina. However, we did not see statistical separation between disease observed in the wheat treated plots compared to the no treatment plots. And throughout this presentation, I'll just be presenting on results from the first field trial. As the second field trial is ongoing, we have strawberries out there right now, so we don't have the disease incidence or the microbiome data yet. So fumigation reduced disease in the first field trial. Summit 515 and WB9229 wheat did not. How about the pathogen in the soil? So I've noted here that the end of season samples with an asterisk are rhizosphere samples, and those first 2 time points are bulk soil samples. So we did not see significant differences between any given treatment in terms of the level of Macrophamina faecelina in the soil. So pathogen inoculum was not significantly reduced by any given treatment. Okay, now going to microbiome. So this chart is showing you bacterial community composition between the different time points and treatments. So you see the squares represent pre-treatment samples, and the squares are all clustered pretty closely together, showing you that the bacterial community composition was pretty similar at first. Then we go into post-treatment samples, which are circles, and you can see that the black, green, and yellow circles, so that's the no treatment control and the wheat samples, are still clustered pretty closely together, but the red circles, so that's the post-treatment fumigated samples, have formed their unique community composition. So we can see a distinct change to the bacterial community that occurred with fumigation. Looking at end of season, so this is those rhizosphere samples, we see that, again, the no treatment and wheat samples are clustered more or less together, but that unique change that had happened to the fumigated samples is remaining in the end of season samples. So we see fumigated and rhizosphere samples show a unique bacterial community composition, and the fumigation treatment effects are long-lasting. So even the end of season samples that were fumigated still have a distinct bacterial community composition. This is the same thing, but for fungi. So again, we see clustering for the most part of pre-treatment samples. We see not too much change with the wheat and no treatment control post-treatment. However, we do see a distinct change in the post-treatment fumigated samples. But as you look at those X's, you see now that the red X's are more incorporated with the other samples. So the fumigated samples at the end of the season fell more in line in terms of fungal community composition with the other samples. So fumigated and rhizosphere samples again show a unique fungal community composition, but this change doesn't last all the way into the end of the season. Okay, so this is looking at beta diversity, and this is showing you on the vertical axes of all of these plots, you're looking at all of the samples from each treatment. On the horizontal axis, you're looking at the same samples. So it's basically if you just were to make a line between any two of the samples and go to the square where they meet, you're looking at how similar those two samples are to each other. And this scale that's given on the right side shows you this metric, which is relative dissimilarity in terms of theta YC. And so the more blue a square is, the less similar those two samples are. The more red, the more similar they are. So first, I want to look at pre-treatment. So you see here, there's not a lot of uniformity, or we can't pull any distinct patterns from it. Same with the end of the season. There's sort of no distinct patterns we can make. However, post-treatment, I want to direct your attention to where the WB and Summit post-treatment samples meet. And you see that that whole block where they overlap is pretty red, showing us that there's a similarity there. So there's a similar bacterial community composition between WB and Summit samples after they've been planted and wheat has grown. We'll contrast that to looking at WB and Summit samples post-treatment compared to the post-treatment fumigated samples. So that's very blue, very dissimilar. And so we could say that there's a lot of differences between the bacterial communities found in fumigated samples post-treatment and the wheat samples post-treatment. So pre-treatment and end of the season bacterial communities are diverse, whereas we see unique bacterial community composition in post-wheat samples. So WB and Summit share a lot of bacterial community. This is the same thing, but for fungal community composition. So again, I'd like to direct your attention to pre-treatment samples. Here we see, with the exception of a couple samples, more red. So that shows us that initially samples were more similar in terms of fungal community composition. End of the season, though, we see a lot more green and blue. So there was a lot more diversity and unique samples in there. Here, this is again the intersection between WB and Summit. It's not quite as red here. So we see, although there is some similarity, some shared fungal communities between post-treatment WB and Summit, it's not as similar as it was for bacteria. And again, we see that fumigation caused a really distinct fungal community composition, as we see that post-treatment fumigated samples are different from not only WB and Summit post-treatment, but also no treatment control post-treatment, as it's all blue. So pre-treatment fungal communities are more uniform, and end-of-season fungal communities are more diverse. Post-wheat and no treatment control samples are both unique from fumigated samples. Moving on, so this is looking at Pseudomonas are what we think might be a key player here. So how did levels of Pseudomonas species change with the different treatments? As you can see, though you're looking at all of the time points and treatments here on this chart, there are a couple samples that stand out, and those are the post-treatment fumigated control and the end of the season fumigated control samples. So those samples had significantly the most Pseudomonas species out of all of the samples. We do see that still the post-treatment WB and Summit samples are significantly higher than the no treatment control at the same time point, but to a lesser extent than those two fumigated samples. It is important to note, though, as you see, I've listed the specific species composition on the legend here on the right. Different species are found, whether it's fumigation or wheat. So in the fumigated samples, you see a lot of Pseudomonas putida and Pseudomonas taiwanensis, whereas in the wheat samples, there's a larger proportion of it is Pseudomonas amygdalae. So fumigation amplifies the overall Pseudomonas species, and wheat does as well, but to a lesser extent compared to the no treatment control. But there's a different species composition there. Finally, I'd just like to acknowledge some other players that we saw here. So we thought that Pseudomonas might be a potential antagonist that could be amplified by wheat. We also saw that there were other bacterial species that were significantly higher in both wheat treatments compared to no treatment control. So all these species you see here were significantly higher in terms of OTUs, or operational taxonomic units, which is the unit of measure we use for microbiome analysis. They were significantly higher in both WB and Summit post-treatment compared to no treatment control. Some of these have been shown to be potential antagonists already in literature. So those that you see in bold belong to a genus that has been described as antagonistic by previous literature. And the ones with the red asterisk are significantly greater at the alpha level of 0.05. So we're even more confident that those are significantly higher. So this is just something to keep in mind. We do see Pseudomonas species here as we expected, but we see lots of others as well. So several fungi antagonizing bacterial genera were in common among post-treatment wheat samples, certain Pseudomonas species included. Yet to come, as I mentioned, the third and final district survey is being conducted by Samantha Simard in Ventura Oxnard this year. Additionally, year 2 of the field trial is ongoing in field 35. So we still have to do plant mortality and microbiome analysis, which I'll do with the help of Cooper Calvin, my fellow graduate student. And Cooper is also looking at wheat, potentially in combination with other strategies such as crop termination, to see if that could have an impact on Macrophyllina root rot. What conclusions can we make from what I've discussed today about the district surveys? So all 4 soilborne pathogens of California strawberry are prevalent. All 4 were found in both districts, between 14 and 52% of diseased fields. And in Watsonville Salinas, as we saw, there was relatively similar prevalence, which sort of goes against the hypothesis that just one had become the most common in the district. Macrophyllina phaseolina, however, is the most prevalent in Santa Maria, as we thought might be the case. And this could be due to weather, irrigation practices, things that might relate to the biology of the pathogen. We also saw root knot nematode in Santa Maria, which we did not expect. So this is something that growers should be on the lookout for in future seasons. How about conclusions from the wheat cover crop trial? So it looks like wheat, as it was used here as a one-time cover crop, does not appear sufficient on its own for management of Macrophyllina root rot. It's important to note, though, that the level of pathogen we have here in field 35 is very high due to past trials that have artificially inoculated the field. So on average, between year 1 and year 2, the average initial pathogen level we saw was 22 CFUs per gram, which is pretty high compared to, if you think about the 2 CFUs per gram we had in the greenhouse trial that was enough to cause plant mortality. And likely grower fields are experiencing slightly lower on average disease pressure. So that's just a caveat I want to make to this study. We also saw that the 2 wheat cultivars we used here, Summit 515 and WB 9229, as well as fumigation, did amplify the Pseudomonas genus in the soil, and the species composition within that varied. There may also be some possible additional antagonists in the microbiome that might be worth exploring in future research. And with that, I would like to say thank you for listening. And this is my new email address, I guess, now. So if you have any further questions, you can contact me there, and I'll also take questions now. So thank you.
Please fill out the form that I put on the table. It's not necessarily evaluating Mary's presentation, but the master's thesis program that we have at College of Agriculture. And please ask questions that you have for Mary.
Marco?
You have a great presentation. So is there like a threshold for like how many CFUs is acceptable in the field, or is there any such thing like that?
Yeah, that's a great question. So I think the CFU. Oh, sorry. Thank you. The question for those of you who are on Zoom was, is there a threshold of macrophamina in the field that's considered acceptable, or like, I guess like the threshold that can cause disease? So we think it's similar to that for verticillium, so somewhere around 2 CFUs per gram. Yeah, I think it's similar to verticillium.
And you're saying that these fields at an average were 22?
So this field trial here was 22. Growers in general, past literature has shown, like within the fields that were sampled for that study, it might be closer to 10 CFUs on average, based on like grower fields that were experiencing die out right from the pathogen. So sort of biased that way, but yeah, somewhere around 10 maybe.
There's a question on Zoom from Dr. Chakudam Muttukali. With your microbiome sequencing data, what soil-borne pathogen genera did you identify in addition to macrophamina, or are these data being analyzed?
Yeah, it's a great question. So if I understand the question correctly, in conducting our microbiome analysis, did we identify any other soil-borne pathogens?
Right.
No, not that I know of. We didn't detect any other soil-borne pathogens to it. Yeah, no, we didn't. Just macrophamina.
And then there's another question from Lindsay Pedonseli. In the Watsonville Salinas sampling, I think you said in about 27% of plants, no pathogen was detected. Do you know what's going on there?
Yeah, great question. So the question was, in the 27% of samples in Watsonville Salinas where we didn't detect any of the big 4 pathogens, what's going on? Why are plants dying? Yeah, so that's something that Cooper is looking into. So he's looking at things like salinity, like so abiotic factors, and also emerging diseases. So things like Pythium, Rhizoctonia, and Neopestalotiopsis, those could be causing die out as well. But yeah, that's a great question, something we're still looking into for sure.
Another question from Samantha Simard. In the soil-borne pathogen survey, did you see any trends regarding particular pathogens becoming prevalent, dominant, exclusively late season or early season?
Okay, so I think the question is, in the surveys, did we notice one pathogen more earlier in the season or another like later? So for both surveys, the sampling occurred over the span of like 3-4 months. So definitely didn't span the entire season. And I don't know that we saw any significant differences in what was prevalent early or late. I know generally, you know, in the time before we sampled in the season, things like Phytophthora and Pythium are more common usually. And then as you go later into the season, Macrophyma, verticillium, fusarium are more common, but we only sort of sampled a snapshot of the season. So I don't think we saw any trends like that.
Another question from Yuchen Wang. Any hypothesis that why Pseudomonas species increased after fumigation treatment?
Yeah, thank you for your question, Yuchen. Hello. The question was why we saw so much amplification of Pseudomonas in the soil following fumigation. So fumigation, as we know, it kills microorganisms in the soil, which releases a lot of nutrients that were in those cells. So this provides an environment for opportunistic microorganisms such as Pseudomonas species to use a lot of those simple carbon compounds that are produced to grow and become prolific in the soil.
Any other questions from the?
You had mentioned in the first field trial that after one of the treatments that there was a difference in like the uniformity versus diverse fungal communities. I'm forgetting exactly what treatment that was, but you had said it was uniform and then there was more diverse. I was wondering if you could elaborate on that, like what that would mean in that context. Like, do you want diversity or do you want uniformity?
Yeah, I think this is worth going back to. So you're probably talking about, so what I was talking about is like, if you look at pre-treatment, is this what you're talking about? You're looking at pre-treatment here, there's more diversity versus here, it's more uniform.
Yeah, I think so.
Okay. Yeah, so I think it's not like better or worse. I don't think for us, it's sort of like just interesting because you would expect like pre-treatment, we're just sampling, right? Like different areas of the field that haven't been treated differently at that point. So you would think they would be, relatively similar to each other. So I'm not sure if it has like implications. I'd have to look into that. But I think just in general, it's interesting to note that in general, there was more uniformity with the fungal community versus the bacterial community started off being pretty diverse, even like pre-treatment. Yeah. Hopefully that answers your question. Yeah. Okay.
I don't know where I was going with that.
Well, that's fine.
I'll think about it.
Okay, think about it.
What's more important, like in the fumigated or in the wheat, where the or the fumigated had that that higher pseudomonas, right?
I don't think so.
No, we're on the separate, but that's.
I was thinking like if a highly diverse fungal community would be better in terms of not causing disease because there would be less of a monopoly on one fungal type.
Yeah, well, yeah, totally.
Competition, so there'd be less possibility for.
Totally. I think, yeah, like bacteria and fungi are both sort of like in the soil competing for like space, nutrients, thing like that. So I think it's like good to look at both. But yeah, if there is like more uniformity, maybe that would create more space for like fungi like Macrophyma. You mean to like, yeah. It's interesting. Evan?
Other than the fumigation, did you see any differences in nutrient composition in the cover crops, and do you think that could have affected the strawberries and their susceptibility?
Yeah, so Evan asked, other than the nutrient effect we saw post fumigation, did we see any nutrient differences or effects in the soil following wheat growth? And yeah, we definitely did. So as we know, cover crops take a lot of nitrogen out of the soil, which we definitely noticed. And in the first trial, we saw at first in the early stages of strawberry growth, smaller strawberry plants where wheat was grown versus the other one. So then we decided to supplement with fertilizer for that. So yeah, definitely cover cropping does introduce other like nutrient effects we have to think about. Yeah. But this year, like we saw that strawberries were planted like pretty close to when we had a bunch of rain. And in the wheat plots, we saw that the plants there weren't so affected by the heavy soil moisture because we think like the root structure of the wheat leftover really helped soil drainage. So like, yeah, like pros and cons, things definitely to think about with management. Yeah.
This is another question on Zoom. Jack Foster is asking, in terms of your disease survey, I find it interesting that Macrophomina was more prevalent in Santa Maria, even though they utilize more drip lines versus Salinas Watsonville districts. How big of a role do you think moisture plays in Macrophomina disease incidence versus other factors? And he also has a question about cover crop, maybe.
Okay, sure. Yeah, thanks for the question, Jack. So Jack's question was that we saw more Macrophomina in Santa Maria, even though they have more drip lines there. So is there something to do with soil moisture there? Yeah, and disease incidence. Yeah, so I was trying to point out, like in Watsonville, they, as you know, they use like 2 rows of plants and 2 drip lines. So that's like a 1-to-1 ratio. Whereas in Santa Maria, it's usually less than 1 because some growers have 4 rows of plants and 3 drip lines. So that's already less. And then some have 4 rows of plants and 2 drip lines. That's even less. I guess some have 3 and 3, but that was rare. So I guess, yeah, like definitely on average, plants in Santa Maria are getting potentially less water than Watsonville. Yeah, and we totally think that might play a role.
And the second part of the question is for your cover crop trial. How do you think growers would feel applying this to their fields, especially with the additional benefit of wheat being a possible non-synthetic nitrogen addition?
Yeah, okay. So the question was, how would growers feel using wheat as a management tool, especially with wheat being a potential non-synthetic nitrogen source?
Mm-hmm.
Yeah, so right. So I mentioned that nitrogen can be taken out of the soil with wheat growth, but if you incorporate that back into the soil, that is then potentially a slow release of nitrogen. That's not synthetic, so not like a synthetic fertilizer. Yeah, I mean, obviously, you know, the whole point of this project was to potentially sell it to growers, right? And so I think it needs some more research in terms of the disease aspect. But from a management, just general perspective, wheat is pretty easy to grow. It's cheap. The seeds are cheap. It doesn't require a long growing season, so you only need to grow it for a month or 2. And then, yeah, if you were to incorporate it, it does have that release of nutrients over the long term. So yeah, we're hopeful that in that way it's pretty easy. And some growers already incorporate wheat or similar crops like triticale or rye for that reason. Yeah.
11-2. Are there any like last minute questions that you have for Mary? If not, let's thank Mary for Thank you everyone for attending, and now we have to have some questions from Mary. You can come. Thank you again.